You rarely take a pure chemical. To safely and reliably treat a disease, a raw active ingredient has to be transformed into a finished medicine. That final product is called a dosage form. It is a carefully engineered mixture where the active drug is combined with specific additives, excipients, that solubilize, preserve, and protect the chemical so it can be manufactured cleanly at scale. The success of this engineering is judged by a strict metric, bioavailability. This measures exactly how fast and to what extent the active drug actually reaches the patient's systemic circulation. Before any active ingredient can achieve its required bioavailability, it has to obey a foundational rule of biopharmaceuticals. Regardless of how it enters the body, a drug substance must be in a liquid solution before it can cross cellular membranes. This map represents the series of chemical environments and biological barriers that a drug must navigate to be effective. Most drugs make the jump from regions like the small intestine into the vascular system through passive diffusion. The molecules are driven strictly by a concentration gradient, moving naturally from areas of high concentration to areas of low concentration. Other molecules cannot diffuse passively. They require specialized carrier-mediated transport mechanisms, where the cellular barrier actively pulls specific compounds across the membrane. A dosage form is a specialized vehicle engineered to outmaneuver these specific biological barriers and deliver a precise, reliable dose to the bloodstream. This timeline illustrates how the chosen route of administration strictly dictates a drug's onset time, forcing the physical form of the medicine to adapt. The oral route is the most common and convenient, but it forces a delayed onset. The medicine has to physically travel through the gastrointestinal tract, break apart, and dissolve before absorption can even begin. As a pill travels, the environment shifts drastically, dropping to a highly acidic pH of 1 in the stomach, before rising to a basic pH of 8 in the large intestine. Because cell membranes are more permeable to unionized molecules, this pH gradient dictates exactly where weak acids versus weak bases can successfully be absorbed. Alternative entry points offer specific tactical advantages. The rectal route allows a drug to enter the systemic circulation while bypassing immediate inactivation by liver enzymes, a hurdle known as the first pass effect. Parenteral routes, like intravenous injections, bypass cellular absorption barriers completely. They deliver liquid solutions directly into the vein for immediate, predictable blood levels. If you take that exact same drug and suspend it in an oily vehicle rather than an aqueous solution, you create a depot preparation. Injected into muscle, the oil releases the drug slowly into the body over weeks. The respiratory route demands even tighter physical parameters. For an inhaled powder or aerosol to reach the deep alveolar zone for rapid absorption, the individual drug particles must measure exactly 1 to 5 micrometers in diameter. The anatomy of the entry point dictates the physical formulation. Scientists have to build the medicine around the specific requirements of the delivery route. To meet these strict anatomical requirements, formulators manipulate the physical properties of the drug particle itself. Physically shattering the drug into smaller pieces, a process called micronizing, exponentially increases its total surface area. For poorly water-soluble drugs, this physical modification is an essential strategy. This equation is the Noyes-Whitney formula. It is the fundamental mathematical law governing how fast a solid drug will dissolve into a surrounding fluid. The A variable represents surface area. The formula proves mathematically why the micronizing process accelerates dissolution. A larger surface area directly increases the rate at which the drug dissolves. The C's represents the drug's inherent solubility, and K is the intrinsic dissolution rate constant. If either of these values is too low, the physical dissolution of the pill becomes the absolute bottleneck preventing absorption. These metrics are so critical that they form the biopharmaceuticals classification system, a global framework used to predict intestinal absorption based strictly on solubility and dissolution data. Once dissolved, the drug still has to permeate the cell wall. The pH partition theory explains that biological membranes are lipophilic, meaning they are fat-loving. They readily permit unionized forms of a drug to pass through, but physically repel highly charged ionized species. A drug's partition coefficient measures this exact lipophilic character. It dictates the molecule's ability to cross the biological membrane and reach the bloodstream. Therapeutic efficacy is bound by these laws of physics and chemistry. If the math of the Noyes-Whitney equation fails, or the membrane repels the molecule, the drug simply leaves the body unabsorbed. Solubility doesn't just depend on particle size. It depends on how the solid powder is structured at the molecular level. Through a phenomenon called polymorphism, identical chemical molecules can pack themselves into entirely different crystal lattice arrangements. These distinct structural arrangements are polymorphs, and they behave like completely different materials. They exhibit radically different dissolution rates, solid-state stabilities, and therapeutic effects. The polypeptide hormone insulin demonstrates polymorphism in clinical practice. An amorphous precipitative insulin contains unstructured microscopic particles that absorb rapidly for short-term effects. But a rhombohedral crystalline form of insulin absorbs slowly, extending the duration of action. Maintaining that precise crystal structure during mass production is incredibly difficult. The intense physical stresses of milling, drying, or extreme mechanical pressure can accidentally trigger a polymorphic transition, ruining the medicine's intended design. Beyond physical pressure, the chemical environment poses a constant threat. Heat, oxygen, and moisture readily degrade active ingredients, causing solvolytic breakdown in compounds like aspirin. Even the additives required to make the pill can be dangerous. Magnesium stearate is a common, highly effective tableting lubricant, but thermal analysis proves it chemically interacts with and degrades aspirin inside the formulation. Formulating a solid dosage form is a delicate balancing act. Scientists must lock the drug into its optimal crystal state and defend it from its own chemical environment throughout the mass production process. All this physical chemistry ultimately serves a single purpose: treating a specific clinical disease in a human patient. A patient experiencing a sudden angina attack needs ultra-rapid systemic relief, relying on a fast-dissolving sublingual tablet placed under the tongue. Chronic conditions, by contrast, require heavily engineered oral tablets that release their doses slowly over hours. Patient demographics also dictate physical design. Infants require highly customizable liquid solutions where the dose is adjusted by volume, whereas adults default to the convenience and stability of compressed solid pills. To ensure a patient actually complies with the treatment, formulators manage organoleptic properties. They utilize synthetic flavors, stable color complexes known as lakes, or water-insoluble derivatives of the drug to mask overwhelmingly bitter tastes. Modern pharmaceutical science is pushing delivery mechanisms further. Polymer therapeutics and nanomedicines bypass general systemic distribution entirely, targeting specific cancer cells directly. Manufacturers utilize crystal engineering, using solvents like supercritical carbon dioxide to fabricate flawless, custom-tuned particle structures. These advanced manufacturing techniques are required to support the rise of biotechnology. Macromolecular proteins, peptides, and viral components require specific formulation strategies to survive the body's natural defenses. Utilizing computational pharmaceutics and in silico modeling, researchers are designing highly sophisticated delivery systems tailored to the specific genetic and metabolic profiles of individual patients. The design of a dosage form represents a synthesis of physical chemistry and biopharmaceutics, the process of engineering a fragile raw chemical into a precise, reliable, and